The main objectives are to learn about the detailed account of the Human Genome Project, to understand about the HapMap project. Introduction. After the advent of recombinant technology and DNA sequencing techniques, the scientists proposed to sequence the entire human genome to understand the causes and treatment of 4 to 6% of genetic diseases. In 1985, Robert Sinsimer proposed the idea of sequencing the human genome, becoming the topic of the worldwide discussion. Followed to this, several scientists supported this idea. Charles Delacy was instrumental in launching the Human Genome Initiative in 1986 and got approved in President Reagan's 1988 budget. James Watson became the first director of this project in 1988 and initiated the groundwork. US Department of Energy and the National Institute of Health in 1990 formulated the goals of the 15 years Human Genome Project with a fund of $3 billion. The HGP has the ultimate purpose of identifying and locating the position of all genes in the human body. In 1993, Francis Collins became the director of the National Center for Human Genome Research. Later, this has been named as National Human Genome Research Institute. The working draft of the Human Genome Project was prepared in 2001. Due to the technological advancement 2 years ahead, the project was completed in 2003. The completion date of Human Genome Project coincided with the 50th anniversary of Watson and Crick's description of the structure of the DNA molecule. Hence, in this episode, the following aspects are going to be emphasized: A. Human Genome Project, B. Working draft of Human Genome Project, and C. HapMap project. A. Human Genome Project. Genome is a total DNA of an organism including genes. Genomics is the study of the total genome of the organism including the structure, function and organization. Each cell in humans consists of a complete set of the genome of 3 billion base pairs. HGP is the international 13 years of collaborative research to sequence and map the human genome that started in 1990 and was completed in 2003. Along with the USA, the other 17 countries have involved in Human Genome Project. They are Australia, Brazil, Canada, China, Denmark, European Union, France, Germany, Israel, Italy, Japan, Korea, Mexico, Netherlands, Russia, Sweden and United Kingdom. The following are the aims of the HGP. Identify all the approximate 1 lakh genes in human DNA. Determine the sequence of the 3 billion base pairs that make up human DNA. Store this information in databases. Improve tools for data analysis and address the legal, ethical and social issues that may arise from the project. Along with the sequencing and mapping of the human genome, this project has also aimed at the genome sequencing of other model organisms like mouse, fruit fly, etc., which have shown importance in the field of science. The model organism help to study the disease conditions and other essential genes. At the beginning of the project preparation, it had made 2 phases for the project. The first phase was a physical mapping of the human genome and DNA sequencing, along with the computer analysis of the sequence information. The second phase, followed from the success of the first phase, includes the sequencing of DNA clones, which are gathered together by the physical map of the genome. The primary resources used for HGP are capillary based DNA sequencing machines, one core optical system concept of Perkin-Elmer 3700 sequencing machine used by Celera and others. 2. The mega-base sequencer was pioneered by Richard Matthews, University of California, Berkeley. Fluorescence dyes for DNA sequencing, bacterial artificial chromosome, that is BAC, superior stability and large size, and supercomputers. The public funded HGP addressed the objectives systematically by isolating chromosomes and fragmenting them. They cloned them to vector and sequence the fragments and finally assemble the pieces using top down and bottom up approaches. After the mapping of human chromosomes, the sequencing was started. In 1988, Craig Venter, a genome expert, started a private company called Celera Genomics to sequence the human genome in 3 years using his methods called shotgun method and supercomputers. The sequencing of the first human chromosome 22 was completed in December 1999, while the sequencing of chromosome 21 was completed in 2000. With the involvement of Bill Clinton, then the President of USA, Craig Venter and Francis Collins have decided to publish the first working draft of the sequence of the human genome in 2001. The information provided in the first draft included a sequence about 2650 megabase of DNA. It was shown that human genome is 25 times larger than the Drosophila and Arabidopsis genome that was sequenced earlier. B. Working draft of human genome project. During the study of human genome before sequencing, researchers were estimated that there could be around 1 lakh of genes. In June 2000, a working draft of the entire human genome was completed. In February 2001, the analysis of the working draft were published. In April 2003, HGP sequencing was completed and the project is declared finished 2 years ahead of schedule. Final version of the complete human genome project includes 99% of gene containing DNA that was announced on April 14, 2003 by Francis Collins, that is 99.99% accuracy. And he said that the sequencing is not completely complete and perhaps never will be. After the sequencing of the human genome, it was found around 30,000 genes. About 40% of the protein found homology with the proteins of Drosophila and C. elegans. About 1278 protein families were predicted from the genes, in which 94 proteins were explicitly shown to vertebrates. Humans have many genes not found in invertebrates. They are antibodies and T cell receptors for antigen, the transplantation antigens of the major histocompatibility complex, cell signaling molecules, including the many types of cytokines. These molecules participate in blood clotting and mediators of apoptosis. The genome consists of about 1.1% of exons, 24% of introns and 75% genome consisting of intergenic DNA. During the formation of mature RNA, the introns are removed by the RNA splicing process. In 70% of intergenic DNA, 44% are made by the transposable elements. It also reduced the number of protein coding genes to 24,500. Other genes may have the functions of producing RNA products, including rRNA, tRNA and miRNA, etc. Some of the milestones achievements of Human Genome Project are: the human genome size is 3 into the 10 to the power of 9 base pairs, equals to 3.2 billion base pairs, which accounts for about 6 feet in a diploid cell. Genes are present in low gene density and vary among chromosomes. For example, on an average of 50 genes per million base pairs, 23 genes per million base pairs on chromosome 19 and only 5 genes per million base pair on chromosome 13. The size of the genes covers a spectrum of sizes 21 base pair to 2400 kb. Dystrophin appears to be the longest gene of 2.4 million base pair with 79 exons. Titin encodes a single polypeptide of 27,000 amino acids. This project also consists of the ethical, legal and social implication program. This ELSI program was started in 1990. The main aim of this program is to identify some issues raised by genomic research and address those issues to the committee of people or society who would be affected. This program mainly concerned in the field of sharing the genetic information and maintaining the privacy, introducing the genetic testing in the area of clinical practice. Ethical issues arise from the genetic research on the people and educating the students, healthcare employees along with the society about the genetics and other issues that arise from the genome research. C. HapMap project. Genomic variations can result from genetic material changes due to deletions, insertions, translocations and duplications. The substitution of a single nucleotide can be seen in AT to GC or GC2AT. This is called single nucleotide polymorphism, SNPs. The variations in the genome include the number of SNPs. These variations make one genome different from other genomes. The SNPs can be present in one population and absent in other populations. Even if it is present in both populations, then it may vary in the frequency. The SNP produced from the single gene mutation in one person can spread through the population. The association between these SNPs can be seen in the population where one SNP may associate with the other SNP that occurred by the mutation in the ancestral chromosome. Some of the SNPs on the DNA can occur close to each other. In this case, the chances of crossing over occur between these SNPs on the chromosome are less. These SNPs may tend to inherit together from parents to the next generation. These polymorphisms on the chromosome may inherit as the genetic blocks. These genetic segments which inherit together through generations are termed as haplotype. The study of these haplotypes can show the relationship between the individual ethnic group and evolution. The study of these SNPs and haplotypes identifies the critical genes whose mutation leads to diseases like breast cancer, glaucoma and rheumatoid arthritis. The association between the SNPs or haplotype are studied to predict the susceptibility of the individual to various diseases. Sometimes it can identify the specific gene associated with the particular disease. Hence, the study of these SNP haplotypes helped to develop an international HapMap project. The International HapMap project was started in October 2002. The international effort was made to build the most detailed map of human genetic variation as a tool for medical research. Organizations involved in the project are the Beijing Genomics Institute, China, the Wellcome Trust Sanger Institute, UK, and the National Human Genome Research Institute, USA, and non-profit biomedical research groups and private companies in Canada, Japan, and Nigeria. This builds upon the International HapMap project, which produced a comprehensive catalogue of human genetic variations. For this project, 270 individuals from 4 populations, namely CEU, that is CEPH, Utah residents with ancestry from Northern and Western Europe, 30 trios, CHB, Han Chinese in Beijing, China, 45 individuals, JPT, Japanese in Tokyo, Japan, 45 individuals, and YRI, Yoruba in Ibadan, Nigeria, that is 30 trios were used. The HapMap catalog helps to identify about 130 SNPs linked to the wide range of common diseases, including type 2 diabetes, coronary artery disease, prostate and breast cancers, rheumatoid arthritis, inflammatory bowel diseases, and several mental illnesses. All the information is collected in this project and made available for further research. To conclude, after the completion of HGP, even more exciting and challenging than the race to sequence the genome, a variety of follow-on projects were launched. Some of them are as follows: ENCODE, Synthetic Genomics, 1000 Genome Project, Personalized Genomics, and Genome Editing. In the next episode, other aspects of HGPs and their implication will be discussed. Thank you.